Hello, everybody. This is topic 7 of Marine Aquatic Ecology course, and this time we're looking at regulated population growth and growth of age-structured populations using life table analysis. So last time we looked at unregulated or unlimited growth of populations, exponential or geometric increase without limit. So this lecture, we're using math to analyze regulated population growth. So we're at the population level of organization, the population unit of this class, and focusing on this part of the struggle for existence, logical structure. We're developing a rigorous mathematical predictive theory of the struggle for existence. So specifically, we're saying populations no longer increase without limit. As populations increase, the amount of resource per individual goes down, which means there's strong selection on individuals to become more competitive for resources and use resources more economically. Okay, in other words, this is Darwin's struggle for existence and natural selection for high competitive ability. So in other words, we have feedback between the population level of organization and the individual organism level of organization. So as density increases, resources become scarce. This has an effect on fitness of individuals. And remember, fitness of individuals defines the growth rate of the population, which then influences the density of the population, which then influences the fitness of individuals, which then determines the growth rate of the population, and so on. So we've got this feedback or regulation going on, regulation on growth rate and population density that controls and prevents the density of the population from rising without limit. So we're talking about population regulation as a result of this negative feedback between populations and organisms. All right. In regulated population growth, we no longer assume that resources are infinite. And we no longer assume that the rate of population growth is determined only by the number of individuals or the density of individuals in the population. Now, resources are finite, and so restrict the rate of population growth in addition to the number of individuals that are there. In fact, there's a relationship between the number of individuals and the amount of resources, and those two together determine the rate of population growth. Okay, so as population size increases, the amount of resource per individual goes down. Therefore, fewer individuals can survive and reproduce, but survival and reproduction determine the rate of population increase. So as populations increase, their rate of increase should go down. So we've been assuming that the rate of increase is proportional to n, population density, so it should always increase. The only way it can go down is if in reality the rate of increase is not proportional to n. So the only way for it to be proportional is if r, the proportionality constant, the little r we've been talking about in last lecture, is in fact a constant, which is what we assumed last lecture. We assumed that r is a constant. So the only way the growth rate can no longer be proportional to n is if the per capita rate of increase, little r, is not constant. In other words, little r is not constant, but decreases as n increases. So let's assume that little r, right here on the y-axis, decreases linearly along a straight line as n increases. So little r is the birth rate minus the death rate. So this must mean either the birth rate goes down or the death rate goes up with increasing n. So here's a plot of little r, the per capita growth rate, against n. And this makes a simple assumption that the decrease of r is linear as n increases. And on top up here, we see the equation for this straight line in terms of the two intercepts. The two intercepts are where the line crosses the y, which is at little r0, and where the line crosses the x-axis, which is this constant k here. So as n increases, this fraction increases, and so the amount that you subtract from 1 increases. Okay, until n is equal to k, then this number in parentheses becomes 0. And when this number in parentheses becomes 0, little r becomes 0 down here. And little r 0 is maximum when n is close to 1. So when n is close to 1 individual, then, or 2 individuals, 1 female, then we have the maximum per capita rate of increase, R0. And then when n increases to k, that becomes 0. So anything in between is along this straight line. Okay, but remember, little r here, the x-axis, is still the per capita growth rate of the population. It's just no longer a constant. So the growth rate of the population is still this little r times n, which is the slope of the population trajectory curve. So we still multiply little r times n to get the population growth rate. And so this is little r right here, so let's multiply it by n to get the population growth rate. Okay, but first, what should this look like, little r times n? Well, for low n, the curve should approximate exponential increase, and it does, approximates an exponential increase right here. Then the break-even point is reached right here, or the inflection point of this curve, and the slope starts to decrease again, and eventually an n is reached as n goes up here on the y-axis. Now we have x on the t-axis here. But as n in this graph increases, little r gets closer and closer to 0 as n approaches k. So as this n approaches k, little r gets closer and closer to 0, so r times n gets closer and closer to 0. So the slope becomes eventually 0. So that means n has reached a point where the rate of growth is 0. All right, so this curve is called the logistic growth curve. Logistic simply means sigmoid or S-shaped curve. Slope increases until it decreases and reaches 0. So this is the logistic growth curve, sigmoid or S-shaped curve. And here's the equation for logistic growth. We start out with the usual assumption, the population growth rate, or the slope, is equal to r times n. But little r is this yellow right here. This yellow part of the equation. And we multiply that by n to get the growth rate or the slope of this trajectory curve. So this is the logistic growth equation right here. The population growth rate or the slope of the trajectory curve is equal to this. It shows that the population increases rapidly at first, close to exponentially. That's because n over k is very small. And the equation then is close to the exponential growth equation, which is just the slope is equal to little r times n. But as n approaches k, this number in the parentheses approaches 0, and the growth then approaches 0. It slows down to 0 when it reaches a size at which r is 0, and this size is k, when n is equal to k. Then this whole product equals 0. All right, K is defined as the carrying capacity of the environment. It's the maximum size that the population is capable of maintaining. And that maximum size, K, is determined by the growth rate of the population. It's the size where the growth rate drops to 0, and the population can no longer increase. So K is the density at which the birth rate equals the death rate. If n ever jumps higher than k, then the number in parentheses here, this number in parentheses, is negative, and the growth rate is actually negative. The population decreases until it reaches k again and holds steady. So little r is the maximum, little r0 is the maximum possible rate of increase, per capita rate of increase, which occurs when n is very small. And little r is called the intrinsic rate of increase. Little r0, is called the intrinsic rate of increase, or the Malthusian parameter, after Thomas Malthus. So this intrinsic rate of increase, or the Malthusian parameter, r0 that we're symbolizing here, is a constant by definition. It's the maximum possible, which is a constant. The maximum possible little r, the maximum possible per capita rate of increase, little r, which occurs at a maximum level of resource per individual, which occurs when the population density is at the lowest possible. So here's the logic again. If the per capita rate of increase, little r, decreases as n increases, then you get this logistic curve down here, which is n plotted against time, where the rate of increase, which is little r times n, goes up for small n, reaches a break-even point midway, and then drops again for larger n, eventually reaches 0 when the population density n is equal to the carrying capacity k. And this reduction in r as n increases has a name. It's called density dependence. So in other words, little r, the per capita rate of increase, is dependent on density of the population. Or an individual's average fitness depends on the density of the population. So in other words, some feature of the environment is reducing little r, but how much it reduces little r depends on the density of the population. It's density dependent. Density dependence is just defined as a change in the survival or fecundity of an individual as a result of the number of individuals in the population, which is the density or number per area. So again, density dependence is defined as a change in the survival or fecundity of an individual as a result of the number of individuals in the population, which is the density or number per area. And why would little r, decrease as n increases? Well, little r is the difference between the per capita birth rate and the per capita death rate. So for it to decrease, the birth rate would have to decrease, or the death rate would have to increase, or both might happen. All right, so if we take into account density dependence, then we get this logistic growth curve or sigmoid-shaped curve. And the equation for the slope of the curve is this. So in the calculus term, this is a differential equation. dn dt, slope is dn dt. And I'm writing slope here so that I don't scare anybody and write dn dt. But this is a differential equation. So this is the slope of the tangent to this line right here. So when n is very small, then the slope is quite low, close to zero. And n increases, we increase the slope to a maximum. And then as n increases some more, it reaches the carrying capacity and the slope becomes zero again. Now you can do some calculus and you can integrate this equation. You can integrate this differential equation to get a relationship between the population density at time t and t. And this is the equation that you get when you do that integration. Okay, so when you fit a logistic growth curve to the data and do this in R or any computer program, you would use this equation. And there are a few different mathematically equivalent versions of this equation, but this is the equation you type in. And then once you fit the data, you'd get values for K, the carrying capacity, values for the starting N, and values for little r, 0, which is the Malthusian parameter. All right, so why do we have a sigmoid curve again? Well, because of density dependence. And because there are factors in the environment that influence fitness depending on density. And those are called density dependent factors. And those are any resource that is consumed at higher total rates at higher population densities. So resources can be food, nutrients, a safe refuge from predators. That's also a resource. Or a refuge from extremes in any kind of environmental conditions. So those references can be consumed because once they're used, there's less available for the next organism. And we can distinguish density-dependent factors from density-independent factors. For example, you can have a factor in the environment that alters the fitness of individuals independently of density. Doesn't matter what the density is. So, for example, a hurricane, or a tornado, or a storm waves, or earthquake, or fire, or landslide. All of those will kill and reduce fitness regardless of the population density. Or just mild variation in any condition, such as a cold winter or a hot summer, can increase mortality no matter what the density is. So it's important to distinguish between density independent factors, which are generally conditions, and density dependent factors, which are generally resources, important resources that influence fitness and can be consumed. Basically, all those factors responsible for the struggle for existence. Many examples of the logistic curve in nature. One of the best marine examples is the gray whale in the Northeast Pacific, the only species of large whale to have completely recovered from overharvest in the 1800s, 19th century, and is now considered to be around carrying capacity, around 26,000 individuals, which was the predicted maximum population size in 2002 from fitting a logistic growth curve to the population trajectory, as you'll do it in lab this week. And it's not known with certainty what the density dependent factors are for the gray whale population and limiting it to this carrying capacity. It could be the feeding grounds, food resources in the northern Arctic region, or it could be habitat for calving in the winter time down in Mexico. The coronavirus total infection curve is also a logistic curve, assuming that the transmission rate stays equal, but of course it doesn't. So we get something similar to, but not quite a logistic curve. So eventually the number of new uninfected hosts or people becomes small, so the virus no longer spreads, and the total number of infected people stays constant. This will eventually happen. And the number of infected people at first is an exponential curve. We have exponential increase and through here. And then it inflects into a logistic curve eventually with a maximum, which we haven't reached yet. That maximum is called the herd immunity, is when herd immunity is reached. Within many plant populations, on land or in the sea, if you plant seeds at higher densities, then the mean size of a plant is smaller, and along with it, its mean number of flowers produced and mean seed set. So this relationship is pretty easy to understand. The amount of nutrients in the soil is enough to support a maximum weight of plants. And as density increases, that maximum weight is distributed over more and more plants, which then have to become each individual smaller. And interestingly, though, the slope of this curve turns out to be around -3/2 in a wide variety of different plant species and growth forms, macroalgae, colonial perennial herbs, trees. Why the slope should be the same across the board is not understood at all, but nevertheless, it's a very useful relationship. In fish populations, for example, Pacific herring, you can plot total biomass, which is a proxy of the population density, against the annual surplus production, which is a proxy for fitness, average fitness in the population, and see that the curve eventually becomes negative as the biomass or density increases. And where it crosses the origin on the y-axis gives you the maximum biomass right here, or the carrying capacity of the populations. And examples of Pacific herring shows different carrying capacities at different locations. Georgia Strait versus west coast of Vancouver Island, for example. And that indicates perhaps different levels of food supply. So this is a pretty common way of showing density dependence in fish populations. A biomass versus surplus production chart. And this could be easily translated into a logistic curve if we wanted to do that. It's just another way of presenting the same kind of data that's used to generate a logistic growth curve. Much lower carrying capacities here out on the Queen Charlotte Islands and the BC Central Coast. Again, the density dependent factors that are causing these differences are not known and the subject of ongoing research. And here is the estimates of the carrying capacities here, just using the results of this curve fit, which you can do in R quite easily, and asking where this curve crosses the y-axis. crosses the y-axis where the biomass at age 3 plus is 88,000 tons. And there are many similar examples of a negative correlation, negative relationship between population density and some correlate of fitness, such as growth rate in individuals. For example, barnacles in this case, semibalanus, balanoides. Growth rate or body mass. And this occurs both among species and within populations of a species. Increased density generally results in lower mass per individual. And this relationship is very predictable and occurs widespread in both plants and animals. All right, here's an example of evidence that density-dependent factors are widespread in the natural world across pretty much all groups of animals. There's a negative relationship between body mass of individuals and the maximum density of individuals per kilometer in populations. So this relationship holds between and within mammals, birds, vertebrate cold-blooded species, aquatic invertebrates, terrestrial invertebrates, across the board. All right, so this leads to a question. Just want to make sure we look at this. If resources are never really infinite, then is exponential growth completely meaningless, like we talked about last time, exponential growth? And the answer is no. Exponential growth occurs to a very good approximation at the early stages of colonization of a new habitat. So for the local example here is colonization of the Mediterranean subtidal by the invasive alga Caulerpa taxifolia. And there are many more examples of colonization of marine habitats by invasive species. Exponential growth also occurs when there's a sudden drop in an important source of mortality, as for instance, a drop in human hunting of gray seals on Sable Island in Nova Scotia in 1960, which resulted in an explosive exponential growth in the number of pups in the gray seal population. Exponential growth also occurs if there's a sudden, very large increase in carrying capacity, as what causes phytoplankton blooms in the springtime. Suddenly, upwelling occurs and there's new resource, and the carrying capacity is much higher, and you can have this explosive phytoplankton bloom. So, in the nearshore and marine environment, this happens predictably in the spring and summer. And the phytoplankton bloom is followed by an exponential explosion of zooplankton that feed on the phytoplankton because their resource and carrying capacity suddenly increased as a result of the phytoplankton bloom. And a well-documented example is the tunicate Thalia democratica. When phytoplankton populations explode, so does the population of the tunicate. And here's the phytoplankton bloom in a Swedish fjord we saw last time. This is a proxy of population density on the log scale, and this is time on the raw scale. So if you plot log scale density against time and get a straight line, this is an exponential curve. If you were to plot this on the raw scale here on the y-axis, it would increase along a curve like this. Okay, and then afterwards, after this bloom, there was a population crash, which often happens after phytoplankton blooms occur because the increased resource was an upwelling pulse, And as soon as that resource gets consumed, there's none left over to support this population anymore. So it crashes until there's another pulse and you get another exponential explosion of phytoplankton. And as we've seen, at the beginning stages of an epidemic, as shown by the coronavirus epidemic in most countries, there is exponential growth. Here's Croatia, exponential growth. And you can get very rapid increase to very high levels as a result of this exponential growth. All right, the prime example of exponential growth under a sudden increase in resources is the human population. So at the Industrial Revolution, resources skyrocketed and mortality rates plunged. As a result, the human population launched a period of extremely rapid exponential growth that continues. Now it's slowing down now, and this has prompted speculation that Darwinian evolution has essentially come to a halt in humans. But sexual selection may still be going on strong. And we can see this in exponential growth by a plot of little r against n. Little r is basically the annual growth rate. R has been roughly constant or actually increasing for the last couple hundred years. But just in the last 15 years or so, R has begun to decrease slowly. And right now it's about equal to 0.005 in the total worldwide human population. And it's still decreasing primarily in Europe and the US, and still constant or increasing slowly in most developing countries. And if we extrapolate the curve of R against time out to the right, eventually it intersects the x-axis or the time axis sometime around the year 2100 at a total N of somewhere around 11 to 12 billion. So in the last few years, the total world population growth rate has been decreasing, meaning we are right at the inflection point of the logistic growth curve. So we're right around here in the inflection point right now, worldwide. So what are the density dependent factors that are resulting in reduction of the human little r per capita rate of increase? Well, that reduction of R is occurring mainly because the per capita birth rate is going down in many countries, most countries probably. And this is related to a wide variety of changes in resources that are important to people, and as a result of that, changes in the economy. So here's an example of a few right here. Okay, so we've looked at three models of population growth. And basically, the first two assume that there's no limit of resources. Geometric is the model used to describe populations where reproduction occurs, say in the spring and summer, when there's an explosion of reproduction and then no reproduction for the rest of the year. Exponential occurs when reproduction is continuous all year round. And logistic growth is growth in which resources are limited. So populations reach a plateau and hold steady there. Okay, and these are the three types of equations used to describe these three types of growth right here. So these are the actual equations relating n to time up here. And this is the differential equation here that relates the slope of the sigmoid curve to the population density. And from this, we can integrate that to get the population trajectory curve like this right here. And remember, this sigmoid growth occurs because there are density-dependent factors in the environment that reduce the per capita rate of increase, reduce the average individual's birth rate, or increase their death rate. All right, now we move to the next subject in the population unit of the course, which is age structure and its role in population growth. So we move on to age structure populations and analyze the effects of that on population growth using what are called life tables or demography. Demography is the analysis of age structure and life tables, basically. And so now we're going to look at the fundamental principles of demography. It's applicable to all species. So up until now, the population was represented by a single number designated as n. So all n individuals we've been assuming are identical for the purpose of this analysis. In reality, however, obviously, individuals always differ. They differ, for example, in their fitness, their ability to survive and reproduce. In particular, young individuals and old individuals are not very likely to reproduce, and very young and old individuals most likely have a much lower survivorship than intermediate aged individuals. And the proportion of young versus old individuals in the population can be very important in determining the rate of increase in the population. So two extreme imaginary examples can make this clear. Suppose all the n individuals in a population were too young to reproduce. Then the rate of increase in the population would be 0 or slightly negative until those individuals grew up to reproductive age. And then it would eventually settle into the expected quantity r times n, little r times n. So now we have exponential growth, little r times n, now that we have reproductive age individuals in the population. Now consider a second population in which all individuals are too old to reproduce. Clearly, the rate of increase in the population is negative. The population will decrease to 0 because none of the individuals are able to reproduce. Post-reproductive age individuals will not contribute anything to the birth rate. They'll contribute only to the death rate of the population. So the more post-reproductive individuals there are in the population, the lower the rate of increase the population will be. And the rate of decrease is determined by the mortality rate of the individuals present. So in any population, the instantaneous growth rate of that population is little r times n. And that's the slope of this curve at this instantaneous moment. But little r is a balance between birth rate and death rate. Births are contributed by intermediate aged individuals, and deaths by very old or very young. All right, so if you look at the lifespan of an individual, these very young contribute only deaths, the very old contribute only deaths to the population trajectory. And intermediate age individuals contribute births. Right, so the slope of the population trajectory, which is the rate of population increase, that's little r times n, which is the birth rate minus the death rate. So birth rate minus the death rate is determined in part by the relative numbers of individuals of these ages. Okay, so if we have a population dominated by old individuals, then the population growth is determined only by the mortality rate. There's no longer any births, so the population has to hold constant or decrease. Whereas if the population is dominated by individuals in the reproductive age, mostly these B individuals here, then there's going to be increase, but very little mortality. And we'll have a very rapidly increasing population. So in order to have a precise estimate of the rate of population growth, we need to know three things. We need to know, one, the age structure of the population, in other words, the proportion of individuals of different ages. Two, we need to know how mortality changes as individuals age. That's called the survivorship schedule. And three, we need to know how fecundity changes as individuals age, and that's the fecundity schedule. Two populations can be identical in any two of these, but if they differ in the third, their growth rate will be different. So how do we figure the growth rate of a population that has a big mixture of individuals of all different ages, all different survivorships, and all different fecundities at different ages? Well, here's how we do it. With a life table constructed as follows. So let's imagine a very simple and straightforward example that will allow us to make these calculations very easily. Let's say we place 1,000 individuals of a nudibranch into an aquarium and follow each individual over its entire lifespan, tallying all deaths and births every month. Remember, nudibranchs are hermaphroditic and have an annual life history. They reproduce and die after 1 year. So normally a life table would keep track of females only, and the sex ratio would allow calculation of male numbers. To make things simple, we just consider a hermaphroditic example, this example here. And so every individual is already female and male, of course. We can put this information in a life table, and that's a life table of Ancula gibbosa nudibranch. On our first column is age. All right, we can break down age in any units we want, whatever is most interesting and most convenient. Let's say for our example that we break down age into 1 month intervals. Age is symbolized in a life table by the letter X. So we start with 1,000 individuals at age 0 or 0 months. And this number is symbolized by N of X, the number alive at age X. What proportion of these 1,000 individuals are still alive at age 0? Well, that's obviously 100%. So the survivorship at age 0 is 1. Survivorship is symbolized by L. L stands for living, and survivorship to age X is symbolized by L of X. So L of X is the proportion of individuals still living at age X, or the proportion that survive from age 0 to X. Obviously, that proportion from age 0 to age 0 is 100%. So the first entry in the survivorship column is always 1. All individuals survive to age 0 by definition. And let's say that at age 1 month, only 800 nudibranchs are left. 200 obviously have died. So what does that give us in our survivorship column? Our survivorship is simply 800 out of 1,000 individuals, or 0.8. And let's say that age 2 months, only 500 remain. This gives a survivorship of 0.5 from age 0 to age 2. And at age 3, only 100 remains, so survivorship is 0.1 from age 0 to age 3. 100 over 1,000 is 0.1. And finally, let's say that at age 4, none remains. So survivorship is now 0 from age 0 to age 4. Notice in the survivorship column that the first number is always 1 and the last number is always 0. You start out with 100% of individuals, you end up with 0%. And we can write down the general equation for this for Lx is equal to n of x divided by n at age 0. The next important piece of information in the survivorship schedule is the proportion of individuals at each age class that don't survive to the next age class, and this is the mortality rate, which is symbolized by m of x, m for mortality. So that's the mortality from x to x plus 1. So what's the mortality rate of each age here? Well, the mortality rate at age 0 is a proportion of these age 0 individuals that won't survive to age 1. What's that proportion? Well, 200 out of 1,000 don't survive, so the mortality rate is 0.2. At age 1, only 500 individuals survive out of the starting 800, so the mortality rate is 500 out of 800. The mortality rate is 300 out of 800, so that's 0.375. And at age 2, only 100 out of the initial 500 survive, giving a mortality rate of 400 over 500, or 0.8. At age 3, none survive to age 4, giving a mortality rate of 1. Now, what equation did we use to get these numbers? Well, it's m of x is equal to 1 minus n at the next age class divided by n at the previous age class. All right, the next column in the life table is the survival rate, which is simply 1 minus the mortality rate. And this is the proportion of individuals of a given age that survive rather than die to the next age. How is this different from our first column? Well, our first column is a survivorship from age 0 to a given age. But our survival rate column is a survivorship just from one age class to the next age class. So we can calculate the total life expectancy at each age class, and this is a more complicated calculation that we don't have time for. All right, now, so this is the survivorship schedule. All these numbers filled out describing survivorship, survival, and mortality. All the information in the survivorship schedule is contained in the N column right here and the age column. This is the number of surviving individuals at the beginning of each age class. All the other columns are computed from this single column. And if all of our individuals did was to survive, then the population wouldn't last very long. We want to calculate the rate of increase. To increase, the individuals also have to reproduce, and they have to reproduce an average of more than a single female offspring. So we need our second set of vital statistics. We need the fecundity schedule. And that's also called the fertility schedule. The fecundity schedule is simply the average number of offspring produced by a female of each age. In our nudibranch, all individuals are hermaphrodites, so they're all females and males. So let's say this is our fecundity column. Fecundity is also called fertility, remember, is at age X. This is denoted by B of X. The B stands for births. So B of X is the number of offspring on average produced per surviving female at age X. Okay, so here are those numbers, 0, 50, 40, and 5, and 0. Now, how do we combine the survivorship schedule and the fecundity schedule to get the rate of increase of the population? Well, the rate of increase is determined by the mean number of surviving offspring produced per female. So remember in our elephant example, each female produced 2 offspring and died. And in that example, the rate of increase of the population was just 2. It doubled every generation. The lambda value for the population was then 2. This means that the per capita rate of increase in units per generation was the natural log of 2, or ln2. So if we know the number of offspring produced per female, that gives us both lambda, which is the increase in the population per generation, and little r, which is the instantaneous increase in the population per unit time. So how do we calculate the mean number of offspring per female overall across all ages? Well, let's just take a simple numerical example using these exact numbers in this life table. Okay, just to make it clear, we'll use these numbers. And let's say we start with 100 females, make it simpler than 1,000. We start with 100 females, and let's say the survival to age 1, the survivorship to age 1 is 0.8. And at age 1, a female produces 50 offspring into the age 0 group. So these 80 females each produce 50 offspring, giving a total of 4,000. Now let's say the survivorship from age 0 to 2 is 0.5. So we have now only 50 females of age 2. And let's assume each female now produces 40 offspring, giving a total of 2,000. Now at age 3, only 10% of the original 100 remain, which is 10. Let's say that each female then produces 5 offspring, giving a total of 50. And then no female survives beyond the age 3. So what is the expected number of offspring per female? It's the total number of offspring produced divided by the total number of females that we started with. The total number of offspring is 400 plus 2,000 plus 50, or 6,050. And the total number of females was 100. So the total number of offspring per female was 6,050 over 100 is 60.5. Okay, and then we can calculate this from the life table just by multiplying the survivorship column by the fecundity column. There we go. Then we add them all up and we get the same number, 60.5. This number is also called the net reproductive rate of an individual and is symbolized by R0. Why do we call it the net reproductive rate rather than the gross? Because it takes into account the mortality of females. It's the net rate of reproduction of surviving females. If all females survive throughout their reproductive period, then the number of offspring would simply be the sum total. In our example, if we calculate this sum total, and it didn't adjust by the death of females before they get a chance to breed, then we'd come up with a much bigger number. It would be 50 plus 40 plus 5 is equal to 95. That would be the gross reproduction rate. Each female would produce 95 offspring rather than 60.5. Okay, so 95 is the gross reproductive rate, or the total fertility of females in the population. It's the maximum reproductive rate possible per female, which she would achieve if her survivorship was 100% across the board during her reproductive period. All right, this net reproductive rate is just another word for lambda. It's the sum of the LXBX column. Two columns multiplied together and then added up. That gives R0, or the net reproductive rate, which is the same as lambda. In our elephant example, each female produced 2 offspring, giving a net reproductive rate of 2.2, which was also lambda. So lambda is just the total number of females produced divided by the total number of females at the start, and it's always assumed per some time unit. Okay, per some time unit. All right, so lambda is the total number of females produced divided by the total number of females at the start. So we use another column in our life table that will allow us to calculate the net reproductive rate. This is the LXBX column. And if we sum all the LXBX terms together, we get the net reproductive rate of an individual in the population, individual female. This is also lambda, this net reproductive rate here, which we've got 60.5. Lambda is the fraction by which the population increases each generation. So in this example, if N1 is the population at generation 1, then 60.5 times N1 is the population density at generation 2. This is R0 times N1, which is equivalent to lambda times N1. Now we can ask, what is the total rate of increase of the population? Well, there are 2 ways to answer this question. If the population growth is discrete with non-overlapping generations, then the rate of increase at generation 1 is lambda minus 1 times N1. This is the slope of the line containing the 2 points at generations 1 and 2. If population growth is continuous with overlapping generations, then we want to know the instantaneous rate of increase at generation 1. This is the slope of the line that is tangent to the curve at generation 1. And the slope of this line, as we saw in the elephant example, is just slope is equal to little r times n, which is the log of lambda times n. And this gives us the rate of increase on a per generation basis, the number of offspring produced per female from one generation to the next. So what we want to know is not necessarily per generation. We want to know for any time unit, per any time unit, the instantaneous growth rate for any time unit. So for example, on a real-time basis, per month or per year. So how can we calculate the rate of increase for every time unit in our example? So the rate of increase every, say, 3 months or 1 month or whatever. Well, we just divide by the length of a generation. How do we calculate the generation time in a life table? Okay, so how do we calculate generation time? Well, there's an easy approximation and a hard way, and we'll just use the easy approximation in this course. So the generation time of any population, or in other words, the number of months or years per generation, it's the weighted mean age of mothers when they give birth, weighted by the number of offspring. And there's a physical interpretation for this. Okay, if you had a ruler represent the age of all individuals in the population, well, the age from 0 to 70 in both directions. And here you have if you plot weights equal to the number of daughters at each age of each female, like a histogram, and the histogram actually is physical weights, then if you want to take that total weight of this histogram and then hang it on the opposite side of the pivot at some age where you get a perfect balance here of this number line. So you've got this weight piled here, and then this weight hanging here, but this weight hanging here is just hanging from a single point. Where would you hang that weight to balance these offspring, at these ages? So this is basically a human example here. Well, you'd hang that weight at whatever point balances here, which for humans is somewhere between age 20 and 30 is the generation time. So if you had 117,000 children here, with mothers from their teens to 50s, and you needed to balance those children from a weight here on the other side of the pivot, then that balance would be at around something between 20 and 30, and that would be the generation time. Okay, there's a simple formula for calculating that number, the mean generation time from a life table, and I'm not going to derive it, just present it. And the formula for the mean generation time is this. It's just basically the generation time equals the sum of all the X's times the survivorships times the fecundities divided by the net reproductive rate from the life table. So to calculate the mean generation time, we just need another column for the table, a column for this big product here. So let's add it, and we multiply all these things together. And this is, we multiply x by lx bx. So 1 times 40 is 40, 2 times 20 is 40, 3 times 0.5 is 1.5. I got 0 here, then we add them all up, that's 81.5. Then we divide that 81.5 by the net reproductive rate, we get to 1.3. That's the generation time for our nudibranch, 1.3 months. So this makes sense from the life table. Most births are between ages 1 and 2. So 1.3 seems to be about right. So what is our instantaneous rate of increase? What's the slope of this curve in real-time units, which in this case would be months? So we divide the natural log of lambda, of the net reproductive rate, divide that by the generation time, 1.3 months, and we get a little r of 3 per month, per individual, per capita. Per capita instantaneous rate of increase. Three females per female every time unit, which is every month. So if n equals 100, the population size is 100, then the instantaneous rate of increase of the population as a whole would be 300 animals per month. That's a pretty big increase rate. So we can use this to calculate our doubling time, or the time t sub d, which is equal to the natural log of 2 divided by little r. So the doubling time of this population is 0.23, about a quarter of a month, or about 1 week. So the population size doubles in size every 0.3 time units, every 7 days. Okay, second, how do we calculate fitness? from a live table. Well, why would we want to calculate fitness? Well, let's say we have two kinds of females in a population that differ in fecundity and survivorship, and we want to know which has the higher fitness. Clearly, the one with the higher fitness is the one expected to produce descendants at a faster rate. In other words, the one with the fastest rate of population increase. If we know the life table for the two females, then we can calculate the total rate of population increase for the two and compare. But to do this, we need to make sure that the rates that we are comparing have the same time units. That's critical. We have to have the same time units. It's going to be number of offspring in both cases, but we want to make sure that the denominator is the same. So let's step back a second and take a quick look at the units of net reproductive rate and fitness and per capita rate of increase. The units of net reproductive rate are per generation. But if we want to compare the fitness of two females, it is not sufficient just to compare their net reproductive rates. Why? Because they might have different generation times. Let's say the net reproductive rate R0 for female A is 2 per generation, and for female B is 3 per generation. Does that mean that the fitness of B is higher than the fitness of A? Not necessarily. This is because the generation time of female A might be far smaller than the generation time for female B. If so, then female A might be able to generate a population of descendants much more rapidly in real time units, days, months, years, than female B. So to compare fitness, we always have to convert net reproductive rate into per unit real time with the same time unit for all females whose fitness we are comparing. And by convention, this is done by calculating little r, the per capita rate of increase of the population per real time. Okay, so here we go. We have two females, female 1 and 2, and they differ in their net reproductive rates. One has 1.2 offspring per generation, and the second is 1.5 offspring per generation. Which has a higher fitness? Well, it has to be female 2, right? She has more offspring per generation. Not so fast. We need to take into account generation time. Of the first is 2 years, the second is 5 years. So let's calculate. Little r for 1, the fitness for 1 is the log of net reproductive rate divided by the generation time, that's 0.09. And little r for the second is 0.08. So even though female 2 has the higher net reproductive rate, she has slightly lower fitness because her generation time is a bit longer, more than twice as long. So to compare fitness, you cannot just look at the net reproductive rate. You have to take into account generation time so that we're not comparing apples and oranges. We have the same denominator in both. It's the same time unit. And also, we're taking into account the effects of age structure. Remember our first diagram, if our nudibranch population consisted of all individuals at age 3, it would be a lot slower in increasing its population size than if it was 100% individuals at age 1. The rate of increase depends on the age structure of the population or the proportion of individuals in each of the 3 age classes. So if the rate of increase depends on the age structure of the population, then what do these numbers mean that we just calculated? I mean, obviously, an R of 3.0 is not going to be true if it's just old individuals in the population, right? Because they're not reproducing. So the lambda that we calculate from these numbers, or the rate of increase that we calculate from these numbers, has to apply to a population that has reproductive individuals in it. So it turns out that the rate of increase we just calculated isn't the rate of increase for any random collection of nudibranch individuals of any age. It's a very special rate of increase represented by this number r, little r. And to see what this means, let's look at a graph of the population trajectory. So this is the population trajectory, namely of size or density versus time on this x-axis. If we start with some random mix of individuals of different ages, how will the population increase? Well, it will jump around at first, then settle into a nice, well-behaved exponential increase curve. The rate of increase we calculated here with the little r is the rate for this end of the curve after it has settled down into its final or equilibrium groove here. We haven't yet figured out how this bumpy portion of the curve is calculated yet. How this portion of the curve looks like depends on the age structure. So what's happening here is that the age structure is jumping around here, and it finally settles into equilibrium here. After it reaches its equilibrium or stable age structure, then it holds constant at that age structure, and the rate of increase then stays constant, and yes, it is equal to what we just calculated here. So when the population settles down, the proportion of individuals of every age is constant from then on. The relative proportions of ages 0, 1, 2, and 3, and so on, no longer changes. So before that happens, all sorts of things are going on here, and we can't use these numbers to say what's going on here. So how and why does this happen, and how do we figure out this portion of the curve here? Well, let's consider it now, how to figure out how the age structure changes from generation to generation. And since this is a bit more complicated than what we just did, I'm going to use an even simpler life table than the one we were just working on, just to make this very easy. So we need to know the age structure. We need to know how survival changes with age, and that's the survivorship schedule, and how fecundity changes with age, and that's the fecundity schedule. All right, so let's figure it out. We're going to put together a new example, simply easier numbers, and we're going to figure this out. So here's the life table. We have ages from 1 to 4 years. And for some reason, I started the table with age 1 here instead of 0. Doesn't really matter. Just means that the fecundity column refers to 1-year-old offspring. And we start with 100 individuals, which gets cut in half each year. Only half the individuals survive to their next birthday. So only 50 out of 100 survive from age 0 to 1, and only 25 out of those 50 survive from age 1 to 2. After that, none survive. So survival is 50% or 0. Very simple. And there's the survivorship column. And the fecundity schedule, also very simple. Each individual produces just 2 offspring except for age 1. And the LXBX column, the sum of this is the net reproductive rate. In this case, it's equal to 1.5 per generation. Okay, and the generation time for this example turns out to be 1.3 years. So what is lambda per year? It is the 1.3 root of 1.5, which turns out to be 1.4 per year. So for this life table at the stable age distribution, we expect that each year the population should increase by a factor of 1.4 each year. So let's see exactly what the population does. Let's start out with a collection of individuals, let them survive and reproduce, and see how the population increases from one generation to the next. So let's say we start out with 10 individuals at age 0, 10 at age 1 year. Sorry, okay. 10 at age 2 years and 0 at age 3 years, since nobody survives to age 3. So this is our population in the first year, 30 individuals total, equally distributed in our 3 possible ages. So what will happen to be what will the population be in the year 2? How many individuals will there be the next year, and what ages will they be? First, let's calculate how many individuals of age 0 will be in the population next year. The only way for individuals of age 0 to be produced is by one of ages 1 and 2. That's these first two here. These each produce 2. So at the stable age distribution in this population, this life table, we expect that each year the population should increase by a factor of 1.4. So let's see exactly what the population does. Let's start out with a collection of individuals, let them survive and reproduce, and see how the population increases from one generation to the next. So let's say we start with 10 individuals in the first age group, 10 in the second, and 10 in the third. And in the fourth, there is none. None survive to this age. So this is our population in the first year, 30 individuals total, equally distributed in our 3 possible ages. So what will the population be in the year 2? How many individuals will there be next year, and what ages will they be? Well, first let's calculate how many individuals of age 0 or age 1, I mean the lowest age group right here. How many of those individuals will be in the population next year? And the only way for individuals in this group to be produced is by those of these ages right here. These females right here are the only ones that produce offspring, only ages that produce offspring. Those offspring will end up in the first age group. Okay, so we've got, so these 10 produce 2 and these 10 produce 2. Giving 20 plus 20 equals 40 newborns in year 2. Now, how many of individuals of age 1 will we have next year? Well, the only way we can get individuals of age 1 is from individuals of the previous age, individuals of the previous year. But these have a survival rate of only 50%. So only half of these 10 survive the next year, giving only 5 individuals of this age in year 2. Same argument applies to the next age. These can only come from age 1, but the survival from age 1 to 2 is 50%. So we got 5 again there. 5 at this age in year 2. And the same for the last age group. We're going to have 0 there. So we have 50 individuals now, in the second year. Now at age 3, okay, so 50 divided by 30 is 1.7. So this is not what we would have predicted. We would have predicted 30 times 1.4 population size in the next year. So we don't get that. That means the population isn't in its stable age distribution yet. And what is our age structure? We started with an equal 3-part mixture of 10, 10, and 10, and the next year the age structure has changed. The new proportions are 80 to 10% to 10%. So let's carry this out another generation. How many in this first age group? Well, we have 5 times 2 plus 5 times 2, giving 20. How many in the next age group? We have 40 divided by 2, or 20. How many in the next? 5 divided by 2, or 2.5. This gives a total of 42.5 individuals, which actually is a drop from 50 the year before. So the lambda from year 2 to 3 is only 0.85. So you can see that the rate of increase is jumping around. We had a drop this year because 80% of the individuals in the population were pre-reproductive. They did not contribute any births, they contributed only deaths. And this continues to happen for about 10 years. We can continue to year 4, and we get 1.5 as the lambda in year 4, and yet a different age distribution. Okay, and we can see this in a plot of age structure versus year. First, we have an equal mix of a third, a third, and a third at each age class. Then the proportions jump around and then gradually settle into the final age structure. This is a stable age structure. In our example, the stable age structure is about 62%, 26%, 11%, and then 0 down here. So once we get our stable age structure, the rate of increase, our lambda, settles down to a constant value also. In this case, it is 1.4 per year. The population ends up increasing by about 40% per year, or by 50% per generation, which is 1.3 year. So this shows a fundamental principle about life tables. If the numbers in a life table stay constant, then the population eventually settles into a stable age distribution and a constant rate of increase, which means it grows exponentially or geometrically over time if there's no density-dependent factors operating. And we have not assumed any for this example. So we have a little jumping around at first. This is a plot of n versus time. And then we settle in by about time 10. We settle into a nice, smooth, exponentially increasing trajectory. And if we calculate lambda each year, we can see the lambda moves back and forth depending on how many non-reproductive and reproductive individuals there are in the population, and settles down into the final lambda value. So these results imply that if there are 5 individuals aged 2 this year, then there will be 5 times lambda individuals aged 2 next year at the stable age distribution. Similarly, if the ratio of 2-year-olds to 1-year-olds is 0.2 this year, then it will be 0.2 next year at the stable age distribution. Okay, and those are the basic calculations that you make from life tables. We have the net reproductive rate, the sum of Lx bx. We have the generation time, the sum of x Lx bx over the net reproductive rate. And we have the instantaneous per capita rate of increase, which is used as a proxy for fitness, natural log of the net reproductive rate divided by the generation time. And we have the doubling time. All right, now for the life tables that we've been looking at today, I've just given you the numbers as if they were just handed to me. In reality, one has to work pretty hard to get the data in the life table. So how does one obtain these data? So this is a common research project, is filling out the data in a life table for some species in nature. Okay, so how do you get these data? In the case of a small, short-lived organisms like the nudibranch we started out with, it's fairly easy to gather the data for a life table. You just simply collect some counted number of very young individuals, then keep an eye on them for a year or so and tally all the deaths as they occur, if you can mark them in some way. And this sort of study gives you what's called a cohort or dynamic life table. Cohort refers to the group of individuals you followed from the beginning. This is a cohort or a group of individuals all the same age. You follow this single cohort over time to get your cohort life table. It's a dynamic life table because you gather the data over a long time period, and you have to be a very dynamic person to do that. This is possible only for organisms with a short lifespan. You can't do this sort of experiment with many organisms. If you wanted to do this with a big, long-lived organism like a sea lion or a whale or seabird, you'd be waiting a long time, decades at least. So for long-lived species, how do you construct a life table? Well, this is called the static or time-specific life table. You can do this sometimes very easily going to a population in nature which contains a mix of individuals of all ages, and then watching that population until a few hundred individuals die and write down the age at which all of those individuals died. This can be done, for example, in many animals that produce annual growth lines in their hard parts. In fish, for example, growth lines are often produced in their scales or their ear bones. In whales, lines in their teeth in odontocetes or ear bones, mystocetes, can be used to estimate their age. In seagrasses, shoot morphology, for example, the plastochrone intervals, can be used to measure age. The simplest method for many species is just to use size as a proxy for age. Like in corals, the colony diameter is a proxy for age. This is often used as a quick measure in fish catch data, for example. So if you can age individuals who died by looking at their skeleton, then you can create a nice life table in this way. And that's called a static or time-specific life table because you gather the data all at the same time. So examples of life tables in the marine environment. Well, in fish, a common way of estimating a survivorship schedule is by plotting length frequency data. So length versus the number of fish with that length in the population. That's a length frequency plot. And consider that the right-hand portion of the curve above the peak is a survivorship or LX curve. This is strictly a size class life table rather than an age class, but there are ways of relating size to age. In corals, colony diameter is a proxy for age of the coral, and the Mediterranean red coral, a size frequency plot is considered to represent the age, size, or age structure of the coral population, or the survivorship schedule of a static life table. Combining the size structure with the fertility or fecundity table results in the overall life table. One then can write R scripts to take into account how the population should change over time as a result of different management strategies, as was done in this paper, strategies that alter mortality components in the life table. So mimicking changes in rates of harvest, for example, and make predictions about whether or how fast populations can recover as a result of these management changes. And this Santangelo paper. So this is the size age structure of a red coral population. These are from colony diameters. So we're looking at the effect of harvest enforcement by rangers and how that affects the population trajectory. So we're looking at increase of the population over time for all these different age classes. And this is the effect of overfishing on the population trajectory over time in all the different age classes. And this is use of diameter. So this is a size frequency plot as a proxy for survival. the survivorship schedule of the coral population. Then we have the fecundity table. Um, calculated as in terms of the number of polyps produced by the coral at different ages. And in seagrasses, similarly, a survivorship schedule can be structured in a static life table that uses plastochrone interval to estimate age, and different schedules can be compared at different locations to make inferences about the effect of the environment on different age-specific mortality rates. So in this example, the mortality rates tend to go up a bit, apparently, with age and at different rates in different locations. And for people, you can put together a static life table for humans by visiting any cemetery and calculating the age of death for each grave, or by conducting a census and asking the age of family members who died since the last census. The human life tables are all static life tables and have been constructed from census data by demographers. And this is one of the purposes of the census, to find the ages at which everyone dies and at which all the women give birth in order to construct the life table. And this information is extremely important to insurance companies who use the census data to figure out their life insurance rates, adjusted according to life expectancy of various groups of people based on sex, age, and race, and so on. And it's also very important to environmental scientists who are working to reduce the negative impacts of the human population explosion, for example, the United Nations Human Population Program. So, what have the demographers of human populations uncovered? Well, here are some statistics about the global human population. So, the exercise and age structure that we performed above in this lecture has been performed for all the major countries in the world by demographers. And here's an example of the United States, the year 2000. These are different ages of females and males, the number of relative population sizes. So here are the baby boomers right here, this big chunk here. There they are at Woodstock, I guess. That's Country Joe, I think. Okay, so you can use these curves to predict how the population structure is changing from, through the decades. You can see these different cohorts moving up to increasing ages over time. And the general rules are increasing populations are dominated by young, and decreasing populations are dominated by old individuals. So Nigeria, for example, is an increasing population dominated by young age groups. Cuba is not an increasing population, not as fast anyway, dominated by older groups, age cohorts. Okay, and you can go through and look at all of these population age structure diagrams. And here we have different regions, population growth, and percent of the population above 60. So South America in the year 2000, population size relative was 515, and then 2100, 100 years later, will be 934, and 33% of the population is over 60. So we have increasing populations here in Africa, South and Central America, slightly in the US. In Europe, most of Europe, it's a decreasing population, and talking about half of the people over 60, roughly constant here in East Asia. And in India, it's a huge population and still increasing. 2100, we expect 35% of the population to be over 60. Population is decreasing in Europe, increasing pretty much everywhere else, except for Japan is decreasing. Then if you look at little r, rates of little r, they're positive in most places, negative in Europe, slightly negative in Europe. And how is little r changing over time? It's decreasing everywhere and pretty much constant in Africa, Central Africa. Constant means exponentially increasing. So constant little r means an exponential increase. So we have exponential increase still going on at the moment in Africa. But every place else, that logistic curve has reached the inflection point and it's starting to, the rate of increase is starting to decrease. Still increasing in Central Africa, the rate of increase is decreasing everywhere else in the world, in the human population. Okay, so looking at life tables allows us to quantify life history strategies in question. We already looked at how you use life tables to calculate fitness. So if you consider the equation for fitness, little r, or fitness equals the log of the sum of Lx bx divided by generation time, then we can see that fitness can be increased by increasing survivorship, by increasing fecundity, or by decreasing the generation time. So now we can use this equation to do a life history optimality analysis where we're looking at the option set and the fitness contours like we were doing in the first unit. And you can do that to make predictions about the life history that maximizes fitness. And the procedure is simple. You can create the option set by writing down equations that relate Lx to Bx to G. And these equations will be guided by your biological knowledge of the species. Then you can create the fitness contours, substitute the above relationships that you figured out into an overall equation for R, for fitness, then plot the contours on a graph in which the two axes are two of the life history parameters you're interested in from the life table. And then you find the combination of Lx, Bx, and G that maximizes R, little r, or fitness. And that maximum is then the prediction of the evolutionary outcome under your mathematical model. So this is the theory that's used to generate those option sets and fitness contours that we talked about before. It comes from population ecology. So we can express the cost of reproduction in terms of the survivorship curves. So here's the survivorship curves. You have age on the x-axis and the log of survivorship on the y-axis. So as age increases, the survivorship might go down and it might be low to begin with. And as the age increases, survivorship might go down, but it might be higher to begin with. Okay, so high cost of reproduction is represented by a survivorship curve of this type right here, in which Lx is high and constant through most of life until survivorship starts to drop. And high survivorship implies high cost of reproduction, as we discussed in the previous lectures. So this is a high cost of reproduction survivorship curve and a low cost of reproduction survivorship curve. Okay, so this is a type 3 here, which represents a low cost of reproduction. It's just the survivorship drops down to very low values at a young age, which implies that there's not much chance of surviving to reproduce later. So your cost of reproduction now is quite small. So these are types 1, 2, and 3 survivorship curves, the log of survivorship plotted against age. So, high cost of reproduction species here, large vertebrates, low cost of reproduction, most invertebrates, algae, and plants. We can also interpret the R to K continuum, or R habitats versus K habitats, and the resulting life history, fast versus slow, in terms of the logistic growth equation, which contains the parameters R and K. These come from the logistic growth equation. So if there are regular density independent factors that decimate the population, then the population is always far from its carrying capacity and will rarely, if ever, reach carrying capacity. This is an R habitat which selects for an R strategy, namely increasing fitness at low density. Increasing fitness at low density means increasing R at low density. Okay, so we can visualize this on this graph here. We have N increasing here up to the carrying capacity. And then you have some density dependent factor decreasing the per capita birth rate as N increases, with the death rate staying the same. We could have the death rate increasing a little bit, but it doesn't really matter. So the difference between these two curves, birth rate minus the death rate, is little r. Okay, so increasing fitness at low density means increasing this little r at low density. And if the birth rate at low density increases while the death rate stays constant, then R will be increased at low density. But there is a trade-off, which means that increasing B at low density might result in a lower B at high density. Okay, so we've got K in this region. Now, if the population is in this region, it's got to be K selected. And if this region is an R selected species. All right, so if we're an R-selected species in this region of this graph, so the N tends to be low because some catastrophe, density independent catastrophe or disaster is keeping the population at low densities, far from its carrying capacity, then fitness will be increased by increasing the birth rate. But there might be a trade-off, so that increasing the birth rate at low density might cause a decreased birth rate at high density. So increasing at low density causes a decrease at high density, all right, which will cause the carrying capacity to move down to the new equilibrium where the two curves cross, the birth rate and the death rate curves cross. And so this is where the two balance out. And so this is the equilibrium population density, the carrying capacity. So reducing the B curve at high density means a reduction in K, the carrying capacity. So this represents an R-selected life history strategy, or a fast strategy, or a low cost of reproduction strategy. R is increased at low density, which increases R0, the maximum R, while K is reduced. So an R-selected population has a high little r0, or high fecundity, but a low carrying capacity. And a K-selected population in a K environment is the opposite end of the spectrum. This is a population that rarely experiences any kind of density independent disturbance up here. So it's a stable population near carrying capacity with scarce resources per individual. So increasing fitness is expected to require strong competitive ability. And if individuals evolve greater competitive ability, it will increase their fitness at high density, which means increasing the distance between the A and B curve, the B and D curves at high density. This is likely to come at a cost of fitness at low density. For example, not enough resources left over to have a high fecundity or a high R0. So K-selected populations are predicted to evolve higher carrying capacity, increasing the curve, the birth rate curve, at a high population size. Increase in little r at high n, which, because of trade-offs, will most likely cause a decrease in little r at low n. But ultimately, what's happening is that you've got a new carrying capacity. You've increased the carrying capacity. So the shape of the survivorship curves is commonly used for a wide range of species as one means to describe their life history strategy. And often, instead of an actual survivorship curve, some proxy is used, such as an age or size frequency distribution curve, as we've seen for Pauli's curves for various fish species. If the mode of the size distribution is a large size, like up here, then Pauli proposes the species a K-selected species. If the mode of the size distribution is a small size, down here, then he proposes the species as an R-selected species. This argument is based on the assumption that size distribution curves have essentially the same shape as life table survivorship curves, Lx versus X. So a nice example of survivorship curve analysis is Tussenbrook's 2002 paper on the seagrass Thalassia, in which is shown that some populations tend to have more K-shaped curves, K-selected curves, like this type 1 curve here, a high survivorship until advancing age happens, and then you have a collapse. And some populations have more intermediate curves, like these type 2 curves, but they could find no Thalassia populations with R-shaped curves. This implies that Thalassia tends toward being a K-selected species with some variation. Okay, let's do a quick review then of the R versus K selection spectrum of life histories and how they relate to the regulated density-dependent population growth and the logistic growth curve. So in a K-selecting environment, this is exactly what Darwin was talking about in the struggle for existence. At populations that are near their carrying capacity, resources are scarce, density-dependent factors reducing fitness are dominant, and there's a strong natural selection for ability to compete for resources, which means for food and space. So if there's variation in and heritability of competitive ability, then the population will evolve towards more efficient harvesting of resources, better competitive ability, and therefore more individuals will be able to coexist together. The population will evolve to a higher density, therefore evolution of greater competitive ability will bring a higher carrying capacity, higher K. But because of trade-offs, inevitable trade-offs in physiology and anatomy and in ability to do things, better competitive ability comes at a cost, and that is lower fecundity. Okay, so that means, well, lower R0 in general. That could be lower fecundity or a longer generation time. Longer generation time might be necessary for individuals to develop better competitive abilities. And at the other extreme, we have R-selecting habitats and species. In this case, populations are kept at low density far from carrying capacity by density independent factors such as storms, damaging wave action, and therefore they evolved to maximize their fitness at low density, which is done by increasing R at low density, which means increasing R0, the intrinsic rate of increase. So they become highly fecund and develop fast to reproductive age. So this also means evolution of a lower generation time. But again, because of trade-offs, inevitably evolution of greater fecundity or higher R0 and faster development means loss of ability to compete for resources at high population density. Therefore, the carrying capacity is reduced. So the cost is a lower carrying capacity. Evolution of higher R0 comes at a cost, lower carrying capacity. So if we bring the logistic equation into this picture, we see the following. So this equation, remember, is an equation for fitness. And this is the exponential part of the equation. And then this is the density dependent part of the equation. So these are the density dependent factors are inside this part of the equation in the parentheses. Okay. So in this equation, there are two parameters, R0 and K. So you can increase fitness, you can increase this equation by increasing one or both of these. You can either increase R0 or you can increase K, or you can increase both to increase your fitness. And if we assume there are trade-offs such that you can't increase both R0 and K at the same time, then you can only increase one or the other. You have to choose between the two. But the choice is clear. In environments where populations tend to remain below their carrying capacity, you'll best increase fitness by increasing R0. And those are the R-selecting species. In environments where the population is always close to carrying capacity, the choice is to increase competitive ability, thereby increasing K. So we have evolution either towards R-selected species or to K-selected species, depending on the environment. And this is also a slow-fast continuum. R-selected species should also benefit by reducing their generation time. This makes it more likely that you will reproduce before some density-independent factor wipes out your population. And the reverse occurs in slow species. Evolution of greater competitive ability at high density takes more time. So we would predict the generation time would be higher in slow species than in fast species, higher in K-selected than in R-selected species. So we have a R habitat in which we have density independent factors keeping the population at low density. So there's not selection for high competitive ability, but rather selection for fast, explosive reproduction as soon as you can before a density independent factor comes in and wipes out your population. And all of this can be expressed in terms of the life table survivorship curves. In that R-selecting habitat, if you allocate your resources into reproduction, fast reproduction, then you're not allocating resources into maintenance and survivorship. Therefore, your survivorship should generally be low in R-selecting species and habitats. Whereas in a K habitat, you're near the carrying capacity all the time, and density-dependent factors are dominant, not density-independent factors. So there's evolution towards higher fitness at higher density, which means evolution towards competing for resources and efficient use of resources, which results in an investment towards maintenance, getting resources and staying alive. which means you have a high survivorship curve. That's the high cost of reproduction curve. Okay, so this is a K-selecting situation, high cost of reproduction, also slow, long generation time to develop those abilities to compete for resources. And this is an R-selecting, low cost of reproduction curve, and also a fast life history, because you want to develop fast, have a short generation time and high fecundity to reproduce before those density independent factors occur and wipe your population out. And the classic book on all of these arguments is this one here by Brian Charlesworth, 1980, Evolution in Age-Structured Populations. Check it out. All right, that's it for today on population growth and regulation. Have a great rest of your day, and we'll see you next time.